So what I thought what I would do is really kind of split this into three parts. First, to talk a little bit about drug development and the regulatory path so that we have some understanding of the context that we're coming into with respect to drug development for COVID, for the COVID-19 pandemic. And then the last part will be really short, but just kind of some thoughts about what this all means for the future. So let's start with talking about drug development and regulatory paths, and also introduce the concept of regulatory science. And by the way, I don't really mind at all if people interrupt me. So if there are questions anytime during the talk, just let me know. So when we talk about drug development, really what we're talking about, why do we develop drugs? And that is because we want to improve the health of the public. And so we develop drugs, and in the context of regulatory science, often we use drug as a shorthand for any product that's regulated, like a vaccine, like the usual drugs that we think of, which would be a pill or a tablet or a capsule or a diagnostic or a device. So any regulated health product, we often just use the drug name as a shorthand for that. So what is drug development? It is really the process which we use to bring a new pharmaceutical product to the market. And usually it starts off with a lead compound that has been identified through the process of drug discovery. And then once you have this lead compound, if we're talking about actual therapeutic drugs, is you then develop it and formulate it in such a way that it can become a drug and you can give it to people and test it. And how we do that is through a process of regulatory science, which is really the science of regulatory decision making, because you can develop drugs, but if they're not approved by a regulatory body, you can't really go anywhere with the drug. So it's really important to understand what is all involved in the process of getting a drug approved. So drug development and regulatory paths and regulatory science and regulatory considerations all really go hand in hand, so that you can actually bring a product to market with very clear indications of what that drug is supposed to be used for, what it does, what are some of the issues you need to be aware of, what are some of the expected adverse events that could happen, what are some of the drug-drug interactions that you need to be careful with. So all of that information is included in the drug label. And of course, we do all of this because we want to have public health goals. We want to maximize the benefit of a treatment while decreasing the safety risk. So the regulatory decision making, then, it's all about the benefit and the risk. So we want the benefit of a new product to outweigh the risk. And we can look at this in many different ways. We want the benefit of the treatment to be, either you prevent death or you prevent some other disease progression event, like a heart attack or increase in tumor size, or lung, tuberculosis, pneumonia, etc. You want to really prevent those kinds of disease progressions with this drug that you're going to give. But the drug obviously will always have some risk, because every drug is a foreign entity that you introduce into a body. And you need to make sure that the risk for any toxicity or other ways or other forms of adverse events aren't bigger than the benefit itself. And it sounds simple, and it sounds like, oh yeah, that makes sense, we can do that. But the more we get into it, the more you will see that this is actually quite a complicated topic. And what makes it even more difficult to sometimes grasp is that we don't really have a formula that says, we can calculate the benefit and we can calculate the risk, and everyone will always get the same answer, because we will see that this is really very context dependent and perspective dependent. So first of all, we need to then, when we develop drugs and put them through the regulatory process, we need to have a clear idea of what is the benefit that we're expecting and what is the risk that we're trying to avoid. And in all of this, then also there's always going to be a degree of uncertainty. So these are the three pillars that I call of regulatory decision making. How much benefit do we need? Do we want? How much risk are we willing to take? And how much uncertainty about either of those are we willing to accept at the time of drug approval? And that can become a very, very fine balancing game. And as I mentioned, it's not always the same for everyone that thinks about it. Janet Woodcock, she is the director of the Center for Drug Evaluation and Research, CDER at the FDA. So she oversees the center that evaluates drugs. There's also a center that evaluates biologics, which includes vaccines. There's a center that evaluates diagnostics and devices. So she's in charge of everything that has to do with drug development at the drugs. And now I'm talking about drugs like therapeutics in the traditional sense. She's currently has been pulled off to become part of Operation Warp Speed. But so she is playing a major role there. But we expect that she will come back to CDER at some point when Operation Warp Speed winds down. But I really like the way she describes it. She says drug development can be described as the progressive reduction of uncertainty about the effects of a molecule in people. So a molecule is the drug entity. It's the API, the active product within the active pharmaceutical ingredient in the drug. Of course, the actual pill will include many other kind of excipients and other substances, but we're really thinking about the molecule, the active pharmaceutical ingredient, and what will be that effect on people. And it can be adverse effects or it can be beneficial effects. But really the process of drug development is the progressive reduction of uncertainty. So we hope that by the time we approve a drug, we are less uncertain. We're more certain about both the benefit and the risk of this new drug in people. But I don't think we ever get to 100% certainty about these. And that's something really important to remember as we think through the COVID experiences that we're all living these days. So when we talk about efficacy versus safety, so benefit versus risk, I would say that efficacy is generally easier to assess and demonstrate than safety because it's usually more quantifiable in the short term, not necessarily always in the long term. But if you're looking at something like decreasing blood pressure or prevention of the tumor size increase, or you're looking to suppress HIV virus load, or you're looking to suppress hepatitis C virus replication, those can all be kind of quantified. The side effects, the safety issues, often don't come up till much later, after long-term use or after a certain number of patients have been treated and exposed to the drug when some of these adverse events show up. So this is from a publication of ours from 2003, but it's a very simple graph, but it really shows how, how much it's supposed to represent how much we know about a drug at the time of approval versus what happens over the drug life cycle. So what we see here in the square, that is what we know in terms of the safety of the drug at the time of approval. We only know those events that happen relatively soon, and we only know about those events that happen relatively frequently. So, events that happen less frequently or events that happen later, we would never really see at the time of drug approval. So we really have this much concrete information at the time of drug approval, but of course the drug will have a long life cycle after that. And a lot of that information about safety and long-term efficacy, we won't really find out till much later. So as I said, benefit risk is not a formula. It is dependent on the context and unmet medical need. For example, think about an Ebola vaccine and what we would know about an Ebola vaccine at the time that you want to implement it. And imagine that you're a healthcare worker in West Africa during the time of a major Ebola outbreak. And what benefit would you insist on? What risk would you be willing to accept, knowing that your risk of exposure to Ebola is very high and that getting disease caused by Ebola would be quite serious? Now, when we're talking about yet another lipid-lowering agent, we would have a totally different attitude, right? Because there are lipid-lowering agents out there. And if we have a new one, we would want to make sure it either had better be better than the ones we already have or safer. We certainly wouldn't accept one that had a lower safety standard. So it's very much dependent on what context you're operating in. And the perception and also the assessment of a benefit risk may vary very much by which perspective you're coming from. Are you a patient that's at risk for getting a certain disease or has already been diagnosed with a certain disease? Or are you a caregiver for such a patient? Are you a regulator or are you a payer? If you're a payer, you want to know that whatever you're reimbursing for actually really works and will actually do more benefit than risk to a patient. If you're a regulator, of course, you want to make sure that whatever you approve should have been approved. And no matter what a regulator does, somebody will always say you were too strict and somebody else will always say you were too lenient. And that is sort of this dilemma fate that regulators have in terms of trying to ensure and promote the public health, but also protect the health of the public. And if you're a patient or caregiver of a patient with a rapidly degenerative disease, for example, neuromuscular, and you know that the downward trend is going to happen very rapidly, and if you know that the drug has a chance to work, you would likely want to expose the patient to that drug because there's at least a chance that something might improve. Whereas if you don't do anything, the patient might die within a year or two. So many of our rare diseases have that kind of profile. So what I'm trying to really impress on you is that there's not a formula and that regulatory decision-making very often involves judgment and interpretation of data. And it's not like you can plug it all into the computer and everyone gets the same answer. It's very, very often that regulators will actually disagree with one another. And experts will disagree with one another. And that is fine, as long as everyone is committed to using the best science on which to base their decision. We should all at least have a common framework of what kind of science we're using to make these decisions. And whether the benefit is high enough or the risk is low enough will be a matter of interpretation and judgment. So this really means that there's a great need for trust. We need to trust those that are making those kinds of decisions. And if we should trust them, they have to show that they're trustworthy, that the process is trustworthy, and that there's the adequate transparency that we need to see. And of course, that is sometimes a problem because the way our systems are set up, there's also a lot of confidentiality issues with respect to what pharmaceutical companies do. But by the time a product comes to market, we need to have all that information and have full transparency about what the benefits and the risk of a drug are. And if that for example, the risks increase once we have more information about how a drug is used on the market, then everyone should be able to know about it. But this is, I think, very, very central and is being illustrated over and over again when you hear all of these political discourses by news and news commentators and political commentators in terms of what's ongoing currently with COVID. So we all agree that we need more benefit than risk, but we can't always all agree what exactly that is and what is the level of evidence we really require, how much uncertainty are we willing to live with. Okay, so now who are the major players in the global regulatory landscape? I just listed some of the agencies here. Of course, in the US, we have the Food and Drug Administration, the FDA. In Europe, we have the European Medicines Agency, and I've listed a few other ones from different countries and regions of the world. And there's some interaction amongst these, but basically in every region and every country has their own sovereignty when it comes to making regulatory decisions. The FDA is a very, very important agency. It regulates up to 25% of the US economy. Many people don't know what we think of just drugs and biologics and vaccines, but it also regulates 80% of consumable products, tobacco, cosmetics, animal drugs, etc. So 25 cents of every dollar you spend has something to do with the FDA processes. Why was the FDA initially established? Well, it was established back in 1906 because people were going around selling sometimes very dangerous products with miraculous claims of miraculous cures for things. And the government realized we better do something and bring some standardization into this to protect people. People, they stopped having people be able to make claims that just simply were not true. And it's an act, it's the law, and every couple of years it's revised. And I won't go into too much detail of this, but there was a major revision in 1938 having to do with safety. The 1980s were very important because that was the era where HIV/AIDS, the epidemic, really started to kind of take over the world, and that drove a lot of innovation in terms of how drugs are developed. And then I want to take you all the way up to 2012, when we had the FDA Safety and Innovation Act, shorthand called FDASIA, and then of course in 2016 under the Obama administration, the 21st Century Cures Act. So the 2012 version and then the 2016 version really built on the 2012 version, really brought in a lot of language that facilitates the incorporation of innovation as we have all of these amazing biotech advances. And facilitates the incorporation of innovation, both in terms of statistical approaches, but also in terms of clinical trial designs, et cetera, into the process. So this is just, you can go online here and check this out. But before the 1938 version, there was a lot of awful stuff going on. And you can see here, check out the video and historical photos, and relive the true and harrowing stories that led to the 1938 version of the Federal Food, Drug, and Cosmetic Acts. And it's pretty shocking when you see what actually was going on. And for example, they had a traveling show that highlighted about the 100 dangerous, deceptive, or worthless products. And the problem was that at that time, the FDA lacked the authority to remove these products from the market. And that's why they revised the act to give the FDA that authority. We talked about the implications of HIV/AIDS, and really what that did was bring about the concept of accelerated approval. Normally, when you develop a drug, you have to go all the way to a clinical endpoint, or we call that a hard endpoint, which is death or progression to a new disease stage. And that can take years. But if we can find a surrogate endpoint or a disease intermediate that happens much quicker, that can predict the clinical endpoint, then the timeline and the number of patients required for that study, the timeline is much shorter and the number of patients is much smaller. And so this is really the basis for accelerated approval, often referred to as subpart H in terms of where in the act that language is placed. And I don't have time to go into that much detail, but just to illustrate how a public health emergency really led to innovation in drug development by allowing sponsors, companies, or manufacturers, whatever you want to call them, to get a drug approved on an accelerated basis based on the virus load of the HIV, rather than waiting till that virus load caused that much immunosuppression to actually lead to a clinical endpoint. And also what really the 2012 and 2016 version of the Act brought in was the patient and the patient voice, and really giving the patient a much bigger role in the whole process of drug development. This is just kind of the Act, the 21st Century Cures Act, that was passed by the 114th Congress, and it shows you who actually sponsored it and the dates when it became public law, etc. And you can go and look at this, but it really has a lot of very good parts in it that have really really supported the kind of innovation that we need, although some of that innovation is really slow in coming. So when we think about the 21st century regulator, I've referred to this before, we really have this kind of, almost fight or sort of this counter activity. One of it is to protect public health. So we want to make sure the drugs are very, very safe. But on the other hand, we want to promote public health. And that means that sometimes we may need to approve something that we don't yet quite know all of the safety risks, but it's so important to have something out there for people. And to promote health, we really want to put that on the market. The problem is once it is on the market, what capability do we have to then pull back should there be too much risk that we hadn't seen at the beginning. So it's always going to be sort of this tension between these two roles. And some people really insist that the FDA should protect, and others really think it should be much more promoting. And they will never be able to satisfy everyone. But again, sort of illustrating that this space where these two functions come together can be very they don't have to be gray, boring gray. That can be actually a very exciting space to be in. And really, these two functions really is what contributes to public health. Just very quickly, some other major players in the global regulatory landscape. We have, of course, all the national and regional authorities that have jurisdiction over their own regions and countries. The WHO plays a large role. They're not a regulatory body, but they have a large role in how approved drugs actually get out to countries and people. And of course, the capacity to regulate. The International Council for Harmonization is a very important body as well. They're there to really harmonize the basic overriding principles of standards for efficacy and safety, etc. So they don't go down into a disease-specific mechanism, but they really just talk about what is an appropriate standards for evidence. It was started off by Europe, Japan, and the US. And now we, of course, have many, many more countries. And I thought it was very exciting that China FDA actually joins, or China as a country joined the ICH in the pursuit of global harmonization in 2017. And that was a big achievement for the China FDA. We also have international health regulations. which provide a legally binding global framework to support national and international programs. And that again is when we have diseases like infectious diseases that spread internationally. And they take urgent actions with respect to acute risks to public health, and also work to strengthen national systems and infrastructure. These are some of the key words here and terms, not only infectious disease like the plague, yellow fever, but also environmental disasters like radionuclear releases or chemical spills. So anything that crosses borders, then this international health regulations comes in, and they can declare a public health emergency of international concern. And why is this important? It is important because it means that it facilitates then more flexible ways of actually getting drugs and diagnostics and vaccines to these sites. So it's a decision that is made. And again, not everyone agrees with how these decisions are made. But let's just have a brief look at the Ebola virus disease spread that we had recently. So this is going back to 2018 and the Ebola spread in the Congo. And the group had met and had said that a national public health emergency of international concern should not be declared at this time. But they remained deeply concerned by the outbreak and emphasized that response activity needed to be intensified. And of course, this was also within a war zone. And the reason why it could be risky to declare something a public health emergency of international concern is that that immediately then brings in severe travel and trade restrictions, which can also be harmful to a country. So again, it's sort of the benefit and risk of doing this at the larger global economic level can be quite profound. However, the outbreak continued, and there were attacks, and you can see here the members of the Congolese military standing guard, it became bad enough that eventually then in July 2019, it was declared a public health emergency of international concern, elevating it to that level where everything goes in there to then try and act on it. And if we go back to the previous pandemic in 2013, 2016 in West Africa, that one had been declared a public health risk to other states and unanimously declared a public health emergency of international concern much earlier on. And in that case, there were over 28,000 cases and over 11,000 deaths, and many experimental treatments were tried. We had evidence that there was efficacy in non-human primate models, but then when the trials were done in West Africa, there was no statistic, they couldn't demonstrate statistically that there was protection. And again, kind of really looking at how do we do research under these emergency situations. And what they had done was they did the studies in the most advanced patients that were already near death, because I think the reasoning went, well, they're near death, so they're more, it's more ethical to put them at risk for something bad that could happen with the drugs rather than people who might not actually go all the way to death. But so we never know, would these have been better if they had been used on less advanced patients. But it really is, these situations can get so complex, and we see that with COVID as well, that so much of the survival depends on the kind of general medical care you get, right? How many, the position, proning the patient and all of these other things that can be done to improve the outcome. But anyway, developing health products during outbreaks can be quite tricky. And of course, we want to have vaccines, drugs, and diagnostics. But again, always the question, benefit, risk, and uncertainty. How much uncertainty are we willing to live with under those particular conditions? And the path for vaccines to prevent Ebola, it requires rigorous preclinical testing with demonstrated protection and appropriate animal models. And all of this had happened, and they were able to proceed to clinical trials in humans. And what happened, and they could demonstrate that there was immunogenicity and protection. But the problem was at the time of the West Africa outbreak, there was no phase 3 trial available, because basically you need cases, right, to evaluate the vaccine efficacy. If you vaccinate people and look at how many got infected and how many didn't, you need to have cases of infection. Otherwise, you can't measure the effectiveness of the vaccine. And so it's difficult to do that sometimes, because sometimes pandemics come and go in certain situations, and these kinds of studies can be very difficult to do. But of course, there are alternative kind of designs that can be used as well. But again, this whole issue about how much confidence do we then have in the efficacy? Do we know about the relationship of the immune marker that we're looking to the efficacy? What actually is the correlate of protection? And of course, what are some of the other factors that we need to look at? And we're going through the same thing again with COVID. Fortunately, the vaccines that are being tested have had such a high efficacy threshold that it is much easier to make decisions because the benefit is so clear. There's also another program, which is the monitored emergency use of unregistered and investigational intervention for Ebola virus disease that was set up again by the WHO to really look at what actually happens with these emergency uses and how are they being used. And it's, of course, emergency use means that there's no proven effective treatment. It's not possible to initiate clinical trials immediately. And but this is a mechanism where data can be gathered sort of in a real world data format to support the science as sort of a way of bringing totality of evidence into the picture. And so this is as of August 12, 2019. Of course, by now we do have a vaccine that is approved, and also treatment that is approved for Ebola that happened in the meantime. So by the time we have the next pandemic, we will be much better set. Now, when we come back to the FDA, we've all heard a lot about the emergency use authorization that has been used for COVID. It was started in September 2006, that program. The then Secretary of DHS, Michael Chertoff, that he declared that the Ebola virus presents a material threat against the United States, sufficient to affect national security. And pursuant to that, the Secretary of HHS declared that the circumstances justify the authorization of emergency use of in vitro diagnostic for detection of Ebola virus. So at that time, there was no existing test, and the FDA then allowed the emergency use of an Ebola test. And of course, at some point later on, in fact, October 10th, 2019, the first rapid diagnostic test for detecting Ebola was actually approved. So emergency use authorization is really just for emergency use where we have much less data, but the sponsor is still asked to complete the whole research program so that we can come to a full approval of the diagnostic. And here just an example of a letter to the Executive Director of Regulatory Affairs at CEPFAID, and basically telling them that this emergency use authorization for the expert Ebola assay has been granted. So this is sort of the end of part one. And the key points really are we have benefit, risk, and uncertainty. Regulators are always kind of battling between promoting and protecting public health. We have learned that advances in science can lead to great innovation, and that emergencies very often foster innovation as well. So let's talk a little bit about what's happening for COVID-19 right now. And so when I say drug development here, again, I'm using drugs as all regulated products more generally. And let's just have a little bit of a deeper look into what an emergency use authorization is. So it is a mechanism to facilitate the availability and use of medical countermeasures, for example, a vaccine, during a public health emergencies. And the FDA allows the use of unapproved medical products to diagnose, treat, or prevent serious or life-threatening diseases or conditions. But the criteria that has to be met is that there's no adequate, approved, and available alternative, which of course, when we have a new pathogen like SARS-CoV-2, we don't have any of that. And so emergency use authorization is certainly justified. But the expectations are that the manufacturers will include a plan for active follow-up for safety, and that the manufacturers also continue the clinical trials to obtain safety and effectiveness information for full approval. So just because a sponsor has received emergency use authorization doesn't mean that they stop the program there. So let's just use vaccines as an example. The most important thing when we talk about drug development is what is the endpoint? And that endpoint really tells us what is the vaccine supposed to do? And that will then translate when we have the label, when we have the drug label for the vaccine or for the pill or the tablet or the capsule, is what is the indication? What is this drug for? And we find out that information when we look at the protocols, which will state the primary and secondary objective. And all of this has to really be preset and very clearly stated ahead of time. And I've been really fascinated by the news reports about how vaccines for COVID have been discussed in the media, because I don't think it's quite clear to everybody what it actually is. But if you want to find out more, you go to clinicaltrials.gov and you say what you want to look for. And in this case, I'm putting in Moderna COVID-2, and you get an NCT number. That's the clinicaltrials.gov protocol number that you can then use to do further searches. So the website, once you get there, will give you a brief summary. In this case, it says the mRNA 1273 vaccine is being developed to prevent COVID-19, the disease resulting from SARS-CoV-2 infection. The study is designed to primarily evaluate the efficacy, safety, and immunogenicity of mRNA 1273 for up to 2 years. And I really want to emphasize here, SARS-CoV-2 is the pathogen and COVID-19 is the disease. I think we've all gotten a little bit lazy. We say, well, so-and-so got COVID, and we mean actually they were SARS-CoV-2 infected, because they may be completely asymptomatic and not actually have any disease. And so I think that is something that is going to come back to bite us, because there may be a lot of confusion out there in terms of what we're actually talking about when we talk about vaccines. So on this website, then you will find the primary outcome measures, and this really is the key part of the protocol. What exactly is being measured here? And the primary outcome measure is number of participants with the first occurrence of COVID-19 starting 14 days after the second dose. Number of participants with adverse events. That's very important for safety. Number of participants with solicited local and systemic adverse reactions, and number of participants with unsolicited adverse reactions. So the primary efficacy endpoint is number of participants with the first occurrence of COVID-19. And COVID-19 here means the disease. And then as a secondary endpoint, they're looking at number of participants with the first occurrence of severe COVID-19. And we don't know here exactly how COVID-19 and severe COVID-19 have been defined. So I can't really tell you much more about that. And then another secondary outpoint is the number of participants with the first occurrence of either COVID-19 or SARS-CoV-2 infection. So we are going to hear more about a number of people that had SARS-CoV-2 infection once we get the full results of the trial. But all we really know right now is that there's this 95% efficacy in terms of reducing the first occurrence of COVID-19. Right, so I hope you can sort of follow that. And this is why it's so important to really use the right terminology for these things so that we're all actually very clear on what is happening. And so what will the indication be based on what's been looked at and what the trial is measuring? I would expect that the label would say this vaccine is indicated to prevent COVID-19 disease in, and then I put in X population. So we'll say something about men and women and age and other, descriptions of the population that this was tested in, people with comorbidities, or was it tested in pregnant women, was it tested in children, et cetera, et cetera. But it really is, at the moment, a vaccine to prevent COVID-19 disease, not to prevent necessarily SARS-CoV-2 infection. And the Pfizer vaccine will be similar to that. And then we have also, in terms of therapeutics, we have I'm just looking at the time here. We have the emergency use authorization for the monoclonal antibodies from Regeneron. And they now have official names, casirivimab and indivimab. That's a combination of monoclonal antibodies that can be administered together for the treatment of mild to moderate COVID-19 in adults and pediatric patients 12 years or older, weighing at least 40 kilograms, with positive results of direct SARS-CoV-2 viral testing, who are at risk for progressing to severe COVID. And so again, if you go on clinicaltrials.gov, you can see here it's got a different NCT number, and it gives you the brief summary. So the primary objectives are to evaluate the safety and tolerability. That's always, very, very important at the very beginning compared to a placebo, and to evaluate the virologic efficacy compared to placebo and reducing the viral shedding. So here we get a glimpse of what is the efficacy endpoint. It's reduction in viral shedding of SARS-CoV-2, which will be measured in nasal swabs. And so that's in phase 1. That will be both safety, tolerability, and efficacy. Phase 2, efficacy becomes more important. And then phase 3 is the full evaluation of clinical efficacy compared to vaccines. And again, here, when we look at the primary outcome measures, there are a lot of safety outcome measures. And then the fourth primary endpoint or outcome measure is time-weighted average change from baseline in viral shedding as measured by reverse transcriptive quantitative PCR in nasopharyngeal swab samples. And it's got some other ones as well. So what are some of the potential ethical challenges going forward? If we have an emergency use authorization for a vaccine for high-risk individuals, how can we complete the randomized phase of the clinical research if some of the patients are going to say, well, I might be on placebo and I can now receive the EUA because I'm over a certain age or I have a comorbidity or whatever. And will patients actually stay in the trial to complete the trial? And what happens when additional vaccines are going to be tested? How can we then test a new vaccine against the placebo what if there is sufficient emergency use authorization of the vaccines for everyone, as they are promising us there will be, but we might even have better vaccines. So how do we, against this emergency use background, then develop additional products? And that can get quite tricky. So the lessons learned here are public-private collaboration, I would say, is essential. It's definitely possible. We can see it can be very productive, and we have so many efficiencies gained. And implications for the future, basically, again, sort of this notion that a forced innovation during the COVID pandemic really is changing how clinical trials are being done. Because at the beginning of the pandemic, many centers had to hold recruitment to clinical trials because everyone was told to stay home. And so you shouldn't go to a clinic unless you had a disease like cancer, something that you were being treated for. But many trials actually stopped for other diseases, stopped recruiting or didn't even start recruiting. And so that has really led to innovation in how clinical trials are executed. We're talking much more now about decentralized clinical trials, patient-centered clinical trials, and of course, innovation and technologies to collect endpoints remotely, including digital technology, and that's very effective. And of course, we have again learned that rapid advancement of innovative biotech technology is possible. In this case, it's the mRNA-based vaccines. It's just amazing to all of us involved in this field how quickly that could be developed and reviewed, and we assume granted emergency use authorization. And we know that the Pfizer vaccine already has that authorization in the UK. But where there's a will, there's a way. And regulation is to promote and protect public health, I think, is alive and well. And I think we will have learned so much from this pandemic that will really revolutionize and sort of have a paradigm shift in drug development going to the future. And with that, I will stop.